Hello, everyone, and welcome to the third week of our study of the cardiovascular system and the engineering solutions to solve some of the problems and issues that we have in the cardiovascular system. This week is the third and last week, and so after this, we'll be moving on to the next and pretty much final topic in terms of the main categories of human tissues and organs that we're looking at, but then we'll have a last bit of the module which will be dedicated to advanced processes and tissue engineering approaches. We probably are also going to kind of have a lecture or two in this third part that comes after this section regarding the brain and soft tissue. And then what we're going to do is we're going to have two labs, as I've mentioned already. The two labs, the first of which is taking place next week, so the 8th week of the semester. Please make sure that you have that in your schedule. You're divided to 3 main groups, like the first lab, and each group within we're going to actually divide you to smaller subgroups to perform the experiments and do the experimental work in those 2 labs. So that's all to come, and we'll have one lecture dedicated to that lab activity. So the 8th week, the Tuesday that we meet, we're going to talk about the lab activity that will happen. The Thursday we'll be talking about the lab activity that has happened on Tuesday, and what you'll have to report upon the completion of that. Okay, let's get back to today's lecture. So as I mentioned, we're wrapping up the cardiovascular system section, and today we're going to talk about aneurysms. We're going to talk about stents again, because they're also used in aneurysms. In the next lecture, and also we're going to talk a little bit about catheters and grafts. We're going to talk about valves, heart valves, in the next lecture. And then we're going to try and conclude and summarize everything. So we've got 2 asynchronous lectures. Please make sure to view those before the 8th week. So during this 7th week, which we won't have any synchronous content or in-person lectures, please make sure to view these videos and be ready for next week. Okay, so without any further ado, let's move on to this lecture's content. So I'm just going to review what we talked about in the in-person, giving you a bit more coherence in terms of the entire fluidic behavior and material considerations. So when we're talking about atherosclerosis or any other lesion that happens within human vascular within the human vascular systems in your blood vessels, we have the growth of the plaque, and we mentioned that that is the initiator for the process. And even after you've administered and deployed a stent, it can then again continue to grow, and what we call it is restenosis, in-stent restenosis. And the first thing that that does is it changes the diameter of your blood vessel. Now, when you're changing the diameter of blood vessel, narrowing it down by a slight bit could have a major effect. So if you remember from the in-person lecture, if you change that radius by 5%, it changes that resistance by about 30%, so the flow rate, or in other words, the resistance also, because these two are related. But basically, as you can see, it's the 4th power, so even a slight change can have a major influence on the resistance and flow rate. What this does in turn is that it changes the flow characteristics, turning it from a laminar to a turbulent flow, changing the Reynolds number. And then what that does is it changes the amount of stress, the flow regime that you experience in that part of the vessel, and therefore the possibility or probability of plaque detachment, which is the worst phase, basically, what we're trying to avoid, because we don't want it to go through the circulation and then clog another region of your vascular system. So on a separate line, this change of diameter also changes the shear stress, which also, as I mentioned, it changes the plaque dynamic, so it can result in a detachment, but directly it influences the corrosion rate of the stent. So the stent properties are now changing as you change the shear stress. If it's a drug-eluting stent, if it's a bioresorbable stent, either the drug distribution and dissolution or the stent degradation can also be influenced by this shear rate. Then you also influence the viscosity, because as we mentioned, blood is a viscoelastic material because of the red blood cells and their density within the blood. It's a dynamic material that doesn't just have the properties of a fluid, it also has the properties of a solid. So it's a viscoelastic, it's got both viscous and elastic properties. And as you change the shear stress, the shear rate, you actually change the viscosity, and that in turn changes the entire thing. It changes the Reynolds number, it changes the amount of shear stress. You can see the viscosity is present both in the resistance and the wall shear stress and the Reynolds number. So this is a complete challenge to actually model. It's a full loop, it's a full feedback loop. So to understand perfectly how this stent is going to survive in the human body, you have to have a very complex model, or you have to do regular visits of the doctor to make sure that everything's working fine and to detect when it's actually in need to be changed. It's a time bomb. At any point, the restenosis region can detach, and then that's it. So yeah, we kind of try to at least give you an idea of how complex things are, although we're not really attempting to solve this problem here in this module, hopefully in another one. Then we talked a little bit about manufacturing stents, and we went all the way from conventional processes where you would cut the sheet and bend it maybe, or maybe do direct machining, all the way to new approaches where we're using 3D printing and deposition of material or conversion of photosensitive material to achieve the final geometry of the stent. And so in terms of stents, really, that sort of concludes our the level of understanding we want to achieve within this module. But we're going to talk a little bit more about stents in this session, and then we'll leave it there. So as I mentioned, the focus of this session is aneurysms, and aneurysms also use stents. So we use stents to basically make sure that the flow doesn't circulate within that aneurysm, because as you remember from when we were looking at the disease, we realized that the circulation of the blood in the aneurysm accelerates and speeds up the growth of the aneurysm, and it just makes it worse and worse. So if we can actually divert the flow, not allowing it to go into the sac, basically, then we can actually start to recover the natural and healthy phenotype. So there are two approaches. In the old days, the main approach was using clips, and these vascular clips that you can see on the left-hand side would clip the aneurysm, stopping any flow from going inside. And the problem was really the rupture risk when you actually apply this, and afterwards you actually have a chance of this clip rupturing the aneurysm. It's a very delicate region of the tissue. The more recent approach is using stents to divert the flow, and basically we call them either a flow diverter or intraluminal flow disruptor. The idea is that they just get the streamlines of the flow to basically carry through, follow through the main vascular branch and not to go in there. And then usually this isn't enough. So what we do is then we go in and fill the aneurysm with a secondary material, which we call a coil. And this coil fills up the aneurysm volume, further blocking the flow from coming through the size of the stents into the aneurysm. But you can still see that you're going to have interstitial flow. So what happens is through time, the tissue there is going to grow. So the coil material is very important. And once you've selected the proper coil material, tissue growth is going to be expedited, and you're going to start blocking that region and turning into a solid, solid piece of tissue that no longer is able to disrupt the flow. So basically, this next approach, this more modern approach, is less invasive, but the problem is that you sometimes have reopening. Okay, it doesn't fully clip that region. As I mentioned, you've got the interstitial flow still, and if the remodeling isn't efficient enough, then you could just have repeat procedures needed. There are a wide variety of material candidates and designs for these coils. Okay, here you can see a good comparison, a table showing the majority of those competitors in terms of aneurysm coils. Most of them are made out of platinum. And the reason for using platinum is, of course, its biocompatibilities. Cells like platinum, they can grow right next to it without it really influencing the tissue. It doesn't get rejected. So it's got that inertness. It's got durability. It's a very strong metal, of course, we know that. And it's got electrical conductivity, and it also doesn't cause issues in terms of radiography. It's got radio opacity. So basically, we've got a really good material there to work with, but then geometrically, these companies have varied the shape and design. For instance, this one here is what we call a delta pack. It's a triangular cross section. The primary winding there is triangular, and then it comes into a tube. The good thing is it allows for flexion. Basically, it allows for lots of room to rotate around that axis and to coil up when you're injecting it into the aneurysm. Stryker is a huge company that actually, sort of pioneers this idea of using a copolymer there that's absorbable. So then, as time passes, this is sort of a dynamic coil. As remodeling happens, this gets absorbed and the volume of it changes. Now you've got designs where it's designed to produce more randomness. Okay, so for instance, where was it? I can't find it here. So both of these, the penumbra models and the random loop design here, they're designed to allow it to coil so easily that it actually produces a random packing. And another approach is really, again, shape memory alloys, and the idea to actually make these expandable scaffolds that promote the thrombus formation or thrombosis. So there's a wide variety of these, and each have their own tiny bit of innovation. We're not going to go over every single one, but these main ones just to give you an idea. What we're really aiming at is, apart from the material properties, as I mentioned, platinum satisfies most of those. We want to have a good random packing. We don't want it to damage the sac, the aneurysm, as it's being injected. So it shouldn't be sharp. It should be very flexible and allow for that deformation and coiling. At the same time, you want it to have those secondary properties, such as being absorbable, dynamically changing its composition, etc., which would be bonuses. Okay, so these are all, various designs to address that. Here's sorry, this is all gone weirdly out of. Okay, let's just. So this is an actual example of it. So you can see the coil in that region, and you can see how tissue is growing around it. Here you can see that same process, and this is a rather new approach which was just published this year. And the idea here is that you kind of similar to 3D printing, where you extrude a filament, they're actually printing into this aneurysm sac here. So they're extruding alginate, and this alginate sort of hydrogel also has these particles in it that are metallic particles. And it's a very clever design of this exposure probe here that actually cures the material in place as it's being added. So here you can see, for example, the microfibers here are injected, and then the UV light is curing them into place to block and cross-link the material in place. So basically, this is an idea of sort of implementing a mesh that has a bit more dynamic behavior. It's got a hydrogel, but at the same time, despite its flexibility and low stiffness, it has the ability to solidify and become a strong structure after you've injected it. So again, looking at stents that are used for aneurysms, this is a video I promised you, and this is how they look at OCT images in terms of the cross-section of the blood vessel. You can move the probe through that particular vessel of interest and map the cross-sectional changes, the geometry of the vessel as it goes through the various regions, including the atherosclerosis region. So this has to be done before you implement. In the majority of the cases where it's not so obvious, this has to be done to give a better idea of how the environment looks like and what stent is proper for that geometrical configuration. This here was showing how most of these aneurysm stents, especially the ones for abdominal aortic aneurysm, AAA, which we mentioned is a common type amongst these different aneurysms, is actually deployed. So the stent in this case is deployed via the common iliac artery. And if you don't know where that is, which I would expect that you don't know because we didn't go over this, it's all the way down the aorta. So down here it divides to the two legs. What they do is they make an incision and they insert it through the common iliac artery right here. And they insert one leg there. They can do the other one from that side, and they would meet each other, just like I mentioned for branched Y-shaped channels. We have sort of a stent kissing scenario where they both meet each other and then they're expanded. Or alternatively, you could go through each of these first and put one region and then put the main one here. Another alternative is if you just do one incision and insert both stents through here, and then go for the main one again through that same incision for the main aortic line. So these are various ways to do that. They're all crimped to allow that flexion during the cycle. When the patient's moving or when the cardiac cycle peaks, they have to have some level of compliance there. And to conclude, really, the various methods to test stents, because as I mentioned before, we're looking at various mechanical properties in stents. One of these most important properties is the elongation. how much can you stretch the stent before it failing, because these usually get contracted during the process. They get crimped, and basically when they're deployed, they actually extend. So we want to know what those safe regimes are to reach from the elastic to the plastic regime. And so another thing that's important is the bending performance, because these have to, of course, bend and deform as the blood vessels also bend and deform. So you want to know what that level is. Does it actually fracture at that point? We don't want the wall of the blood vessel to be damaged due to that. And another mode is actually compression here. We want to know the loop strength. This here is a longitudinal strength. This is the loop strength. We want to know how strong it is under compression. When does it burst? Because then you have compression of the blood vessel, you have the inflation of the blood vessel whenever you're looking at the high pressure peaks, and depending on where this is deployed, these could each have increased levels of importance. And here you're actually stretching it also using this specially designed fixture. It's notable that even for the stretching, as you can see, you cannot do it with a simple clamping feature because it's a very delicate structure. So instead of that, we actually have a support tube, and they're clamped using a very nice mechanism there, held from these cylindrical jaws, stretched from that condition. Torsion is another important test, because there are cases where they're going to be under torsion, both in the deployment phase and inside the body. So this sort of concludes our understanding of stents, their materials that are used, the coils that are used in conjunction with them with aneurysms, and the testing of stents. Really trying to give you a background idea of all of this. Continuing on, I'm going to talk a little bit about grafts, and we'll conclude this session. So artificial grafts and biological grafts are the two categories. Artificial, we usually use, again, a combination of polymers and fibers that are both compatible to endothelial cell growth within, and also its integration with the external tissue from the outside. So what happens is usually these are allowed to stay in blood for a bit, so that they're actually so it's immediately before we do the surgery. They're all now submerged, and so when we put it in the body, it's not dry, it's already now hydrophilic to that extent, so the flow doesn't actually prime it for the first time. It's primed outside the body. They're commonly used for larger vessels, not for the smaller ones, and the idea is that you can then suture the vessel in that region where you're cutting or removing something. For instance, you're removing an aneurysm. You use these grafts to actually then connect the two ends of the vessel in that region that's now removed. You want to bridge the gap, so you use these grafts. Usually it's around 5 years. So the patency, as you can see here, the graph shows about majority of them really after a matter of 5 years are no longer useful. Some do a little bit better. Some are a little bit worse. But again, typically around the 5 to 6 year mark, they have to then change them. But by that time, we're hoping that regeneration and remodeling has already gone through its course of action. In the biological case, what we do is we use a vein from the leg, and so we can sacrifice one vein for a region that we need it for. So we've got 2 veins in each leg. One can be sacrificed. That's the most common place we actually use as a spare. And so we can then implant that to the arterial position of interest. And then gradually, as I mentioned, veins and arteries vary a lot in terms of their thickness of the wall, their composition. So this vein has to gradually change its phenotype and start to look more like the artery, and it does. It sort of starts to remodel into that new artery phenotype. So again, as I mentioned, the material that we use here is usually a composite. So in this case, you have polycaprolactone, PCL, or silicon nanofibers on the outside, and then PCL collagen, which is really good in terms of endothelial growth in the inside. And so they enable that endothelial cell growth and spreading on the surfaces, especially because they've got these directional sort of deposition routines, the way we actually produce these. Either we use processes like electrospinning or bulk fiber entanglements, and what happens is that they have directionality, so that allows the cells to sort of gain that initial shape and geometry due to the fiber organization. And then they also have a better mechanical strength because of this composite design. And then gradually the various tissue structures are going to form on the internal surfaces and the external surfaces, but usually the internal. And so where they use these, for example, here I've put an image of when they do cannulation in the extracorporeal life support systems, when you're connecting the system outside of the body to support the heart or the lungs to the inside. You actually use these grafts, and they, although here it looks very messy, they're able to carry flow very efficiently. The sutures work beautifully, and so they're very good to connect that circuitry in the surgical theatre in lots of these various procedures. And here you can see the same material pretty much used in the epithelium rather than the endothelium, so in the skin. And we'll talk a little bit more about this in the next section, but yeah, skin grafts are a very good example of use of grafts. Grafts in general refer to the broader category of epithelial or endothelial replacement biomedical materials and devices. So here we kind of see one example that's used for vascular structures. Okay, so there we're going to conclude it here for today. And in the next lecture, we're going to talk about these guys, heart valves and their replacement. So this is actually a heart valve replacement you saw in the in-person session as well. We're going to talk about various types of these. We're going to talk about how they function, what the materials are, and what their pros and cons are. So you get up to date with what's happening there. And that will conclude our lecture series on the cardiovascular system. Thank you very much for listening, and look forward to see you again in the next lecture.